What's up, guys? This is the MERG. Today it's all about the cell membrane. You can think of it as the skin of the cell. Now we're going to talk about the three main components that make up the cell membrane. We'll add a little more about its characteristics at the end. The first and usually most abundant part of the cell membrane is the phospholipids. This weird alien you see right here in front of you is called a phospholipid. The round portion is called a phosphate head. It's polar, meaning that it's hydrophilic or water-loving. Attached at the bottom are a pair of fatty acid tails. These chains are the exact opposite of its head counterpart in nature. They are nonpolar, meaning they're hydrophobic. A term we use less often is lipophilic. They repel water. One other thing, a glycerol backbone is what stands in between the two of these. It's the glue that holds the head and tails together. Overall, we describe the phospholipid as amphiphilic. It means having both polar and nonpolar properties. Being amphiphilic is super important because it leads the way to how the cell membrane is formed. I'll explain. Let's take a look at this cross-section of the cell membrane regarding only phospholipids. Remember, there are two more very important components we'll discuss about this later on in the lesson. Here you can see that the polarity keeps the heads of the phospholipids together away from the hydrophobic tails. When we look under a microscope, we'll notice that the interior of the cell is polar, as well as the environment that the cell lives in. It all falls into place when you realize that a second layer forms as the membrane, but upside down. Here, you can see how the polar sides naturally gravitate towards the polar environments, both inside and outside of the cell. What's left is this extremely hydrophobic, nonpolar center of fatty acid tails. This double-sided phospholipid structure is the very reason scientists term the cell membrane a phospholipid bilayer. How convenient! Let's move on to our next major component of the membrane, cholesterol. Cholesterol is usually wedged in between phospholipids, like this. They play a vastly important role in stability, basically adjusting the environment and keeping the membrane together. When it's cold, the phospholipids would bundle up tight. This could be a huge problem with mobility of the membrane, and so the cholesterol works to keep these phospholipids from being too rigid. When it's too hot, phospholipids begin to spread apart. Cholesterol becomes the glue to keep them from separating completely. The final major component are proteins. There are several types of proteins that live on or within or even in between the cell membrane. Integral proteins, or what some people may call transmembrane proteins, can be found running through the cell membrane. These proteins provide passageway for the cell to take in and release certain substances. Peripheral proteins are just that. They reside on the peripheral of the membrane. They can be found in any combination, even residing on top of an integral protein. They can act as an enzyme and provide support to the cell shape. Another component worth mentioning is carbohydrates that branch out on the membrane. They can be used as identification of the cell. They act as receptors for cell signaling and play a large role in communications. When carbohydrates are attached to the cell membrane, we refer to them as the prefix glyco. So when you find one attached to a protein, you would call it a glycoprotein. And if you were to find one bonded to a phospholipid, we would call them glycolipids. With all that in mind, the cell membrane has a very convenient property that keeps it alive. It's called being semi-permeable, meaning that the membrane allows certain substances to pass through it, but not others. Yes, even without the need for an integral protein for transport capabilities. We call this property passive transport, or otherwise known as passive diffusion. Passive transport is a spontaneous process where molecules will move from a more concentrated environment to a less concentrated environment without the use of energy. In the membrane's case, that means that these molecules are able to move in and out the cell freely. Let's familiarize ourselves with a few of what can come in and what can't. There's a general way to guess how substances will behave. The cell membrane generally prefers smaller substances. This being because the phospholipids aren't tightly packed. They actually move around freely. The cell membrane also prefers non-polar substances. This is because the center cross-section of the phospholipid bilayer is heavily concentrated with non-polar hydrophobic tails. Remember, this is not always true, but in most cases, it works. Some gases, like oxygen and carbon dioxide, are small enough and non-polar, meaning that they could pass through. Water, H2O, is also small, allowing it to pass through the cell membrane as well. But because it's polar, it will do so at a much slower pace than the gases mentioned. In some cases, there are also larger molecules, like benzene, that are also able to pass through the barrier because they are nonpolar. There are, of course, the ones that have no chance at passive diffusion. Take glucose, for example. It's large and polar. It has to be transported through the cell membrane by transmembrane proteins. Another difficult to pass substance are ions and charged molecules. Charged elements like sodium, which is positive, and negatively charged elements like chloride. One last thing. Let's take a microscopic aerial view of the cell membrane. As you can see, it sort of looks like a painting. In fact, this style of art is also known as mosaic. And since these components of the cell move freely amongst each other, like a crowd of people in a subway, we consider it fluid. Scientists ultimately gave this name the fluid mosaic model. Well, that's it for now. Join me again soon. And if you enjoyed the video, please like, subscribe, and share. Bye!